Hello, I am Dr. Elena Paili, PhD. I work in the field of Alzheimer disease. I would like to discuss with you Alzheimer disease and what we did in order to improve the situation with Alzheimer disease. Currently, there is no diagnostic test, laboratory diagnostic test, there is no treatment, there is no prevention for this disease available. There are 4 million patients were several years ago only in United States of America. Now it's more than 5 million. So for several years the amount of patients dramatically increased, but the cause of disease is still unknown. So only symptomatic treatment is available. Many people think that Alzheimer's disease is a genetic disease, so it's related to heredity, but it's absolutely not true, so it's not correct. The majority of cases, that like more than 90% of cases, certainly not linked to any genetic mutation. So more than 90% of cases of Alzheimer's disease are metabolic or some another reason disease and not genetic. So we're interested, our project focus and our interest focus on that majority of cases of more than 90%. So in order to understand how to treat disease, how to make a diagnosis of disease, what I mean by diagnosis, I mean that current diagnosis is based only on clinical observation, like a conversational diagnosis. It's not a laboratory diagnosis. There is no lab test. So it means there is no test to confirm the diagnosis or to say that this is the wrong diagnosis. It's the first problem. So of course there is no treatment and no prevention because there is no even cause of disease. And in that case, where is no cause of disease, it's very difficult to find the correct treatment. So this is why we started our project from the model of diseases. So we created cell model and animal model. What it means? We took human cells in cell culture and reproduce disease. So they make like mimic disease. We make something like similar on cell culture, human cell culture, and then using mice. We didn't introduce any gene mutation in our model. So we have a metabolic model based on intoxication with toxin. So we found a neurotoxin that induced characteristical manifestations similar to Alzheimer's disease in the brain of mice and in the cultured human neurons. After we found this neurotoxin, we decided to investigate how we can get this toxin in high amount, this neurotoxin. Because generally, this toxin is a normal compound of our biochemical pathway. So we have this neurotoxin, but a comparatively low amount. But it could be increased. What is the way to increase this? One of the way is to get it from food, to get from plant, or to get it from bacteria, because some bacteria produce this neurotoxin. So we decided to check human samples of patients with Alzheimer and healthy people if they contain, if the samples contain some markers of bacteria, specific markers for specific bacteria that produce neurotoxin. In order to check it, we used a non-invasive test. So it means we didn't make biopsy, we didn't take a piece of brain, we didn't take a piece of spinal cord or skin or anything like what we need, or it could be painful to take. It's a non-invasive test. We tested stool. stool feces, so it's absolutely not dangerous for people. So we use stool of normal people, healthy people, and sick with Alzheimer's disease. Clinically approved Alzheimer's disease. And we found difference using our specific markers for potential product of bacteria. We found a difference between healthy people and patients with Alzheimer's disease. So now we have neurotoxin that can induce manifestation in mouse brain, in human cells and culture. We found marker in stool that reflects presence of bacteria in human body with Alzheimer's disease. So it means that we are on the way to make a reliable diagnostic test. And as a result of this test, we can suggest a reliable treatment against bacteria, like to use antibiotics against specific bacteria or to create new generation of antibiotics against these bacteria. So we are going to expand our research to test more samples of patients and more samples of control people that are not demonstrate manifestation of the disease. So this is our way to suggest you new hope in treatment of Alzheimer's disease. Thank you.